Diagnosis of COVID-19 can be done by two methods. One is PCR, that is polymerase chain reaction, and another one is rapid blood testing kits. So first we'll discuss about the PCR. Now to perform the PCR, first we need to collect the sample, and sample is collected from the nasopharyngeal and oropharyngeal swab. Now during this step, collecting a good sample is very crucial, because if we do not collect a proper sample, then there won't be enough virus load on that sample, and that can lead to a false negative result. So collecting a good sample is very much required. Now if the result comes negative and we still suspect the patients for COVID-19, then the sample will be collected from the lower respiratory tract. It includes endotracheal sample and bronchoalveolar lavage, right? Now the technique or the PCR technique which is used for the COVID-19 is called as a reverse transcriptase polymerase chain reaction. Now why it is called as a reverse transcriptase PCR? Because we know that the PCR can amplify, PCR can replicate the DNA into the more copies, right? But our sample contains the RNA. So first we need to convert the RNA into the DNA. So it's a reverse process, right? Normally क्या होता है, DNA में से RNA बनता है, but here we are converting the RNA into the DNA. And the enzyme which is required for that is called as a reverse transcriptase. So from that, this process is called as a reverse transcriptase polymerase chain reaction, right? Now, so in the first step, we'll be mixing all virus sample with the free nucleotides and reverse transcriptase enzyme, so that reverse transcriptase will consider the single-stranded RNA which is present in the sample as a complementary strand and will synthesize the double-stranded DNA. So first step will be the conversion of single-stranded RNA to double-stranded DNA by enzyme called reverse transcriptase, right? Okay. Now after this step, the remaining process will be same as in the other PCR. So let's discuss the polymerase chain reaction. Okay. So PCR, or we can say polymerase chain reaction, is a method of molecular biology which converts the specific DNA into a millions or billions of copies, right? So scientists take a small amount of DNA and then they amplifies it into a large enough amount so they can study it into a detail, right? Now this PCR technique is used for the diagnosis of infections, and apart from that it is also used for the crime investigations, right? हम जो भी fingerprinting and all crime movies में देखते हैं, that is done by the PCR. Then it is also used for the genetic research and molecular biology. Now the instrument which is used in this PCR technique is called as a thermal cycler, right? The sample, or we can say the components that are added into this thermal cycler includes DNA templates, Taq polymerase, primers, nucleotides, right? Now DNA template is nothing but a segment of our original DNA which is going to be replicated. Now Taq polymerase is the enzyme which is required for the DNA replications, right? Taq polymerase is thermostable enzyme. This enzyme is derived from the heat tolerated bacteria called Thermus aquaticus. Then primers. Primers are the specific short sequence of nucleotides, right, which will be used as a starting point for the DNA replications. They helps in identification of specific DNA region or specific target DNA which we are going to replicate, right? Then nucleotides are the building blocks for the DNA synthesis process, right? So all these components will be added to the thermocycler, right, for the processing of PCR. The PCR process includes 3 steps. First is the denaturation, second is the annealing, and third one is the extension. right? So let's discuss each step one by one. So first is the denaturation. So in the denaturation process, the sample is heated to a 96 degrees Celsius, right? So because of this high temperature, right, the DNA sample will break down into a two separate single strand DNA, right? Our original double stranded DNA will get separated into a two separate strand DNA by this process of denaturation. Okay, now the next step is annealing. The annealing word comes from the metallic industry, where the metal is heated to a high temperature and then it is cooled down to remove the defaults or to remove the internal defects in the metal, right? The same method is used in our PCR also, that is why it is called as a annealing. Now in this annealing process, our previously heated sample is cooled down to a 55 degrees Celsius. Now this cooler temperature, this 55 degrees Celsius, will allow the primers to attach to a specific region of DNA, or we can say to a target region of DNA, which will act as a starting point for the DNA replication, right? So that was annealing. Now the next step is extension. In the extension, the sample is again heated to a 72 degrees Celsius, and the enzyme Taq polymerase is highly active at 72 degrees Celsius. So the Taq polymerase will get attached to a primers, it will grab some free nucleotides and will assemble the new DNA strand, right? So at the end of this extension process, our single stranded to separate DNA strands will get converted into a double stranded to DNA, right? So now this PCR cycles, it takes approximately around 10 minutes. So at the end of each cycles, there will be a double number of DNA, right? So if we say that at the end of first cycle there will be a 2 DNA, then at the end of second cycle there will be a 4 DNA, then at the end of third cycle there will be a 8 DNA. So there will be an exponential rise in the DNA numbers. So after 5 to 6 hours we can create billions of DNA copies from one specific DNA. So if you have liked this video tutorial, then don't forget to subscribe and like my videos. Thank you.